Hi, my name is Monica Dayao. I am a fourth-year PhD candidate at Carnegie Mellon University, and today I'm presenting my poster in collaboration with Enable Medicine on deriving spatial features from in situ proteomics imaging to enhance cancer survival analysis. The data I use here is highly multiplex spatial proteomics imaging data, which is essentially images of tissue at single cell resolution, for which each cell you can visualize the expression of tens of proteins from anywhere from 30 to 70-ish proteins at once. And this type of data has the potential to give unprecedented insight into disease states. However, currently methods to predict clinical outcomes such as survival from this data is currently lacking. And so in this work, we wanted to look and see, we want to investigate what features from this data can we extract that are predictive of survival. And more specifically, can we use methods such as spatial statistics to investigate this? And how do these features correlate with survival outcomes in cancer? And so the data that we're using is a head and neck squamous cell carcinoma data set, in which we have 281 samples from 81 patients, almost 2 million cells divided up into different batches. 39 proteins here are measured, and there are 16 labeled cell types. Now, moving on to the methods, we wanted to be able to extract features from this data that use the spatial information. And so we do this in two ways. The first way is what we refer to as neighborhood matrix, in which we take our sample, we do cell segmentation and classification of cell types. And then for each cell, we look at its 10 nearest neighbors and look at the proportion of cell types that are within that 10 nearest neighbors. And then do this for all of the cells in the image. And then this outputs a neighborhood matrix in which we have a 16 by 16 or cell type by cell type matrix. And each element of the matrix corresponds to the fraction of cell types of that column within 10 nearest neighbors of that row, of that cell type row. And so we have a neighborhood matrix for each sample. Another metric we computed is what we call the transformed Ripley's K function. And so for each cell, we look, instead of K nearest neighbors, we draw a radius of R around each cell. And we compute two K functions. These are spatial statistics equations. The first is the weighted K function, in which we sum the biomarker expression within the radius. So that's biomarker expression. So this is done for each biomarker, so each protein is what I mean. And then the unweighted K function, in which we count the number of cells within this radius. And so we actually subtract these two functions, and this produces the metric that we're using. And so for each biomarker, for each sample, we have this function of R. And we only take values at R equals 30 and R equals 80 for our analysis. And so once we have these spatial features, we input them into a survival model. We use a random survival forest, which then outputs a risk score for each sample based on after it's trained. And then we do some model evaluation. So we compare how these feature sets do to non-spatial feature sets. And then we also look at which spatial features are associated with higher survival risk in patients, or higher mortality risk in patients, lower survival. And the main thing that we found is that spatial features do improve predictive performance here. And so in the middle graph, the orange bars correspond to non-spatial features. So these are kind of like our baseline methods. And then the green bars correspond to the neighborhood matrix and K function that I mentioned earlier. And the blue bar is actually combining the neighborhood matrix and K function to input to our survival model. The three gray bars are comparison methods that previous works have done. And we're measuring here the concordance index, which is a value between 0 and 1 that indicates how well the survival model is performing using the respective feature sets. And so here we see that the combination of the neighborhood matrix and the K function perform best. And even though the K function on its own does not perform well, the combination still performs better. And so lastly, we wanted to look at whether the random survival forest could tell us whether certain spatial features have enriched interactions, for example, in higher risk patients. And so what we did is that we separated patients based on the output risk score of the random survival forest, and then looked at the differences in these features between those cohorts. And what we found is that there's an enriched macrophage fibroblast and immune cell fibroblast interactions in the high risk cohort versus the low risk cohort. And so this work is actually being continued by our collaborators, experimental collaborators, to confirm these findings and to learn more about this disease.